I am here, so let's go ahead and resume. I'm assuming that everyone else is here as well. So we left off at the end of the discussants reports on the 3 evidence streams. So now it's time for committee discussion, which could include comments from the members of the committee who haven't spoken yet, questions for the discussants, and so on. So I'm going to go ahead and start with one comment which pertains to the human studies of cancer.
Dana, do we have public comments in here sometime?
Yes, they come after the
it will be. Dr. Loomis, I wonder if also you want you to ask for some follow-up, and staff indicated they were going to follow up. And anytime you would like staff to clarify on those 2 points that they were going to bring back to you, they can do that.
Right, okay. If they're ready to do that now, we could take that up before we go to committee discussion now.
Okay, thank you, Dr. Loomis. I can report back to Dr. Lamero and Dr. Eastman and the committee regarding the 2 questions. The first question from Dr. Lamero is on the thyroid hormone effects in animals. So I'll answer your question based on summaries from OEHA's proposed public health goal draft document for PFOS. The data available include those summarized by US EPA 2016 and more recent data. So US EPA 2016 identified several studies reporting thyroid effects. In general, there was a reduction in free thyroxine or T4 and total T4 levels without significant change in TSH or thyroid stimulating hormone. The effects was consistently observed in rats. So these were rat studies. Specifically in pregnant rats, PFOS reduced T4 and trialdothyronine or T3 levels in the dams and T4 in the pups. In monkeys, there was a significant decrease in total T3 and T4 levels in females only, with no change in TSH. So the newer data since USEP 2016 include NTP 2019 and the monkey study. NTP 2019 reported decreases in T3, free T4, and total T4 in both sexes of SD rats. And the monkey study showed a slight reduction in serum total T4 in both sexes, no significant changes in TSH or free T4. So these are the thyroid hormone effects in animals. Regarding Dr. Eastman's question on the range for the historical control values, so I'll start by saying concurrent laboratory controls are always the most appropriate direct comparison with an experimental group. The historical control data we included in the hazard identification document were not from the same laboratory as the Tomford study, but from other control SD rats from oral studies used by Charles River Laboratories. So the Tomford 2002 study started in 1998 and lasted 2 years. Therefore, we reported historical data from 1995 to 2002, which means these studies were conducted within 2 to 3 years of the Tomford study. Specifically, for the pancreatic islet cell adenoma, the range was 0 to 25.7% in male rats, and pancreatic islet cell carcinoma range was 0 to 14% in male rats. So as you've observed, the value in the control was higher than the average, but it is still within the historical control range. For the thyroid follicular cell tumors in female rats, these are rare tumors. So the range for adenoma is 0 to 1.16%. And the range for the thyroid follicular cell carcinoma is 0 to 0.6%. So these are my answers. Thank you.
Thanks. I'm wondering if you could do that for the liver tumors as well.
Liver tumors, the liver hepatocellular adenoma in male rats range is 0 to 8%. The liver hepatocellular carcinoma range is 0 to 6%. And in female rats, the range for liver hepatocellular adenoma is 0 to 0.29%. And the hepatocellular carcinoma in female rats ranges 0 to 0.71%.
Good, thank you for that. Now, let's move on to committee discussion. And I did have one additional comment that I wanted to offer about the human cancer studies. This is with respect to the studies of breast cancer in which serum PFOS was measured before diagnosis. So I mentioned a concern about those studies in that despite the long half-life, there is some loss of that material. And typically, the exposure is only measured at one point in time. And so what this means is that there's some unpredictable measurement error in that information. So not knowing any more about it, my first suspicion would be that since it's essentially random measurement error, it's likely to result in bias toward the null in that entire group of studies. So that's something to keep in mind in thinking about, you know, the mix of positive and negative results that we see in some studies, so that when we see positive results, likely to mean that those are studies that may have had better exposure information than the ones that have more equivocal results, weaker associations, and so on. So that was my comment about that. So now let's go ahead with discussion with other members of the committee who have questions or comments to offer, and we can try to do what we did before and use the raise hand function. If Dr. Martyr is still watching, maybe she can help me. And
absolutely, Dr. Loomis.
Thanks. And just let me know if I miss anybody. I'll try to watch, but I can't see all the speakers at once on my screen.
Dr. Crespi has her hand raised.
Go ahead, Dr. Crespi. We can't hear
Here we
you.
go. Trying to get off of mute there. Yeah, I just want to first thank OEHA staff for putting together a very comprehensive report. So I really appreciate their work. And I had a question that was directed to the members of our committee and maybe also OEHA staff regarding the animal studies and some of the comments by 3M. In particular, their comment was arguing that the liver tumors in rats are basically an observation, are due to a mechanism of action that's not relevant in humans. So I wondered whether there was anybody who could speak to that evidence one way or the other, and to that particular comment. Should we discount that finding of liver tumors in rats because that biological mechanism is not something that operates in humans?
Thanks. Does anybody like to answer that question? Dr. Eastman?
Hi, I appreciate that. What they're referring to is mechanisms in which tumors are induced, liver tumors are induced in rodents through these nuclear receptor mediated mechanisms. And they tend to be the PPAR, alpha and gamma, or the CAR or PXR. And the challenge is that may be true, but they really haven't provided enough evidence to substantiate that. That's the problem I have with it. I think that may be the case, but usually there's follow-up mechanistic studies to demonstrate that's what's happening, and they don't seem to have done that, or at least I couldn't see that. So that's the way I interpret that.
Okay, anybody else want to contribute to that question? All right, I think Dr. Reynolds had her hand up for a question as well.
Actually, I was actually going to make a comment. I comment a little bit further on the epi-evidence. I can do that later if you want to stick with questions right now, or
Well, just go ahead. I think we'll take it all.
I wanted to definitely add my thanks to the OEHA staff for their always impressive work in putting together these hazard identification documents and providing us with the original articles. I also did want to acknowledge some informative comments from the public, which I thought were quite helpful. There is no question that although population levels are declining, there's been a widespread and continuing exposure to this group of compounds. And this is, we've come to a chemical where actually, finally, we do have some epidemiologic research. Often we do not when we do these reviews. I appreciated that Dr. Stern and Loomis gave a nice review of the epi data, and although limited, the cancer studies really have focused on breast cancer outcomes. It's been a topic of some interest to me, and probably this research is focused on breast cancer because of the endocrine disrupting properties attributed to PFOS, and that it's one of the most common cancers in women. So as articulately discussed by my colleagues, as well as from public comments, the evidence from these studies is most certainly mixed for a number of reasons intrinsic to the challenges of conducting such human health studies. Much of the evidence does come from case control studies in which exposures based on a single blood sample taken at or subsequent to diagnosis of the cases. And since we know little or nothing about the metabolic effects of disease progression or treatment on measured levels, this leads to the associated potential as discussed for reverse causation or difficulties in interpretation in general. Similarly, biospecimens were obtained at various points in time, reflecting some fairly strong secular differences in exposure. And as I'm glad Dr. Stern pointed out, few studies, including my own study, focused on important windows of susceptibility during the life course. I think there has nonetheless been some provocative evidence from the epi literature for risk associations by breast cancer subtype, both premenopausal versus postmenopausal, and also for ER positive versus negative tumors, suggesting that the risk may not be entirely null. And just to share, I do want to mention that our own rather null study in California actually saw a non-significant positive association with PFOS for hormone receptor positive breast cancers, in contrast to an also non-significant inverse association for ER and PR negative tumors, suggesting that there might be some very differential effects. Also, there are a couple studies that were published this year that weren't included in the document that we might want to just consider. One was a fairly large hospital-based case control study from Japan that was 400 cases with blood collected at diagnosis in the early 2000s and 400 controls. And that's the only study, as far as I know, to examine risk associations for linear and branched isomers separately. So while that study found an inverse association for PFOS with most models, they also intriguingly noted the opposite effects for linear versus branched isomers for another PFOS, for perfluorotridecanonic acid, if I pronounced it correctly. Also not included in the HID is a small case control study published in July of this year from Manila with a suggested elevation in breast cancer risk for PFOS, although it was no longer statistically significant when adjusting for a variety of covariates. So as blood was collected in the mid to late 1990s in the French study, which is the study that both reviewers found most informative, probably both because of prospective blood collection and also for the assessment by hormone receptor subtypes, the levels of PFOS were quite a bit higher than in some of the more recently conducted studies. It's interesting to note also that positive results were reported in that study and in the two, albeit small studies of populations that reflected high levels of environmental exposure, the Greenlandic, Inuit, and women in the heavily industrialized area of Manila. Nonetheless, as Dr. Eastman says, the human data are muddled, and I would appreciate a little more discussion from my colleagues in terms of the key characteristics. They were somewhat generously summarized by Dr. Zhang for perhaps 4 of the 8 as being supportive in terms of mechanisms of risk. And I'm still not entirely clear about the lack of consensus from the animal literature, which is also a bit mixed. Nice to know it's not only the epi literature that gets very mixed results. So I just wanted to add those few comments.
Thank you for those comments. Let's see whether the colleagues who reviewed the key characteristics would want to respond to any of those questions. And I have a question for them as well. So maybe they could take both of these at the same time. So in my mind, strong mechanistic evidence usually means that we have evidence from studies of exposed humans. And my impression is that there are not very many of those studies, but I didn't go through and count. So I'd like to hear your thoughts about what makes the evidence strong if it's true that there aren't very many studies in exposed humans.
I can jump in and kind of go back and just highlight some of those that are focused solely on humans, if you like. Okay, so let me just scroll to the top here. Okay, so I think we all agreed that there wasn't much going on with genotoxic, so I'm going to skip that. For epigenetics, I suggested it was limited to perhaps strong, and that was based on the fact that there were a number of studies of DNA methylation in humans with PFOS exposure assessment. One that got brought up was the recent firefighters study that Dr. Zhang shared with us. And that one I noticed in particular, sorry, I lost my place momentarily, that they did some specific work on DNA methylation of a certain gene called RAD1. This is an important checkpoint gene. I thought that was relevant because we had those consistent changes in the position of the cells in the cell cycle. So consistent with this idea that you could have estrogen receptor mediated proliferation that was driving more cell cycle synthesis is kind of like a synonym, if you will, for that. What you have is these molecular signals in the cell where we go through the cell cycle. If there's a damage or problem that arises, these checkpoint genes basically slow down the cycle so that that damage can be addressed either by repair or cell death or other processes. And so RAD1 having differential methylation status with respect to PFOS in this human cohort is interesting. It's also perhaps relevant to the group because there's immunohistochemistry evidence that it's able to be a significant prognostic indicator for both liver and breast cancer in humans, looking at like the protein atlas, which is a repository of data of this nature. The other DNA methylation data that I brought up was 2 studies in cord blood, one based in Japan and one based in Taiwan, that both found DNA methylation increased in association with PFOS for the gene CYP2E1, the gene SMAD3, and a gene called SLC17A9. And SMAD3 is basically a gene that regulates other genes involved in a process that's been shown to relate to metastasis of breast cancer to the lung. So again, something relevant to the breast in that instance. The other epigenetic data that I mentioned that came from a human study was the microRNA, but it was not as human direct in the sense that the only microRNA that was altered by PFOS in a human context was in 2 human neuroblastoma cell lines, but I didn't find evidence that that microRNA 22 was associated with cancer in my cursory analysis of the general literature. So moving on to oxidative stress, where I believe all 3 of us indicated that we thought there was strong evidence for oxidative stress. That data comes from the evidence for 8-oxo-DG. So when you have oxidative stress to a cell, one of the consequences can be that these reactive oxygen species are able to bind macromolecules, including DNA. And so 8-oxo-DG is basically an adduct that forms on DNA that's a marker for oxidative stress. And there was 3 human observational studies that looked at that in association with PFOS exposure. And 2 out of those 3 studies found a dose-dependent relationship between the PFOS exposure and the 8-oxo-DG adduct in those people. One of the studies was in 60-year-old plus people, 126 of them. The other study that was significant with that dose-response relationship was in 597 people of the ages 22 to 63 years old. And the third study that was null was in Taiwanese people from 12 to 30 years of age at a number of 848. And then that was an addict that was also the only other study that looked for that type of addict looked in lettuce seedlings of all things, but they also found it was increased there. There was some evidence of reactive oxygen species also in a study of Chinese newborns where they looked at plasma from cord blood and 581, and they saw increased evidence of oxidative stress as well. So quite a number of humans with the PFOS exposure associated with markers of oxidative stress there. And then the receptor mediated was the other one that got brought up as a strong. And I think that there was agreement that that was mostly on ER. So as you recall, there's quite a lot of it. So let me just skim it real quick to make sure I don't overlook some of the key parts. I think there was, you know, for what it's worth, various 4 different studies that have modeled human ER in silico using what we know about the crystal structure to predict PFOS binding to the human estrogen receptor alpha. And that appears to be possible that it activates through a number of different human cell lines. So not in an observation study, but they've shown that the ER alpha can be activated, as in it can cause promoter activity in human transfected kidney cells, as well as human breast cancer cells. And yeah, I think that's right. And then we also saw proliferation of human breast epithelial cells that was dependent on the ER, as evidenced by using a selective antagonist of ER called ICI 182-780. So I think probably if you want to just stay tight and focused, those would be some of the key human-related outcomes in the receptor-related section. And then I had mentioned KC10 as strong, really as an extension of the ER. The other 2 reviewers didn't mention that, but this idea of increased proliferation seen across rodents and human cells. So they did the human fetal hepatocyte, human ovarian granulosa, tumor cell lines, several of them, and then the increased occurrence of human cells being in S phase, or the proliferation synthesis phase. And that again was the human fetal hepatocyte model and the human mammary epithelial cell model, and then the human epithelial cancer cell model. So I think that probably captures it pretty well.
Thanks, that helps a lot with my question. see whether anybody else from the group that reviewed those studies in detail wants to jump in on these questions.
I'll jump in.
Reynolds,
Peggy, did that address your questions?
that was what I was going to ask as well.
So, I mean,
I think so. I mean, I'm hearing slightly different takes on the whole thing. I think that these key characteristics are very critical in terms of thinking about the sort of mechanistic likelihood of risk associations that we can't necessarily delineate from the human health studies. So it certainly is a complex area, and there were sort of lots of issues that were out on the table. So I'm having trouble getting sort of a sense from each of you about how enthusiastic you feel about key characteristics.
so if I can weigh in, I think of these as sort of, it's a framework by which we evaluate sort of the overall evidence picture, but these are all possibilities. So there's evidence for all these possibilities, but at this point, we don't have a compelling case for any of them. You know, we can say there's a little more evidence for this possibility or that possibility. What you'd like to be able to say mechanistically is PFOS does this initially, and that leads to this second, and then this, and this, and this. And then you can put together, this is a plausible mechanism for carcinogenesis, but I don't see that. We have lots of possibilities. There's evidence for lots of these possibilities, but it hasn't been pulled together in my mind into a really coherent argument.
I hear you. Thank you.
I just want to acknowledge I forgot to highlight the strong immunosuppressive aspects of things when I went through the summary. Looping, I don't know if you want to highlight a couple of the key human studies that were involved in that. I didn't get into as deep of a dive on that since there was so much material.
Yes, I think just following what, okay, Michelle, I'll get back to the cases in just in a minute. So let me follow in what David just said. I think there was a thing, we have a little bit of information here, there, we put them together. I think that's a very good question in order to explain the potential, carcinogenic pathways. So if we all agree, number one, oxidative stress, it looks like a PFOS can induce that pretty strongly. And also see, but we maybe not, but from oxidative stress, you generate reactive oxygen radicals that also can link to the inflammation, right? So I'm just trying to see if how we could have, one KC linked to another one. And so for the chronic inflammation, the KC6 didn't, I think Michelle, you did really, summarize that. But I also want to say, OIHA staff did a really, really good job to put all the different type of the study on the chronic inflammation in that, I forgot which appendix H or something. I think that's a very informative summary. But even though if you look at the study chronic inflammation, it's a very difficult. I think this is a pretty difficult marker to trying to define acute or chronic inflammation when you look at the marker. But I think the data we have so far, at least I still thinking the chronic inflammation, although we still think it's limited, but what we do see the positive supporting evidence from all human, animal, and the in vitro studies. So this is back to from oxidative, how can link to the next to the inflammation. And also we know the inflammation linked to the immunosuppression or immune response. So from that pathway, I think this few cases they actually linked to see if you can think about using the AOP or using current AC idea to form the potential pathways. One thing also I just want to address what Dana was just asking. If you want to make one of the cases, anything strong has to be human. But I think from the ARAC preamble, so it's human, doesn't have to be human in vivo exposure. So if the evidence is a human in vitro or ex vivo data evidence, that still could count as the strong. So at least I think what we have included oxidative stress, immunosuppression, and receptor mediation effects. So this is, I think that's all three had evidence from the human studies. I think plus the epigenetics, 4 of them as well. So back to the immunosuppressive, that's what Michelle wanted me to address on. And actually, somehow I don't find it. I wanted to give a good example, but I think what I have seen here is included one study on the cultured human blood cells, to look at the NK cell activity. So and other studies in mice. But if anybody can back me up, I actually thought we should have some. Do we have any in human? I'm just really trying to look for in human in vivo studies, but looks like there is one in the documents about the CD4 and the CD8 T cells exposure is only in vitro, but I thought I did see some studies in human cells as well, but it's not coming to me. I can look a little bit more, or maybe you have staff can help me here and see if we have more human in vitro data for the immunosuppression suppression. So I just end here for now.
Great, thank you for all those clarifications. I see Dr. Stern has a hand up. Let's go to her question.
Yeah, just a quick comment. So to follow up on Dr. Chang's discussion. So I saw a paper that I don't know, I haven't heard if you included it because it's pretty recent. I think it came out in October of this year. It's by Emir out of University of Illinois. And I don't know if you consider that one because that was a study where they look at prostate cancer cell lines and xenograft models, and they exposed them to PFOS and also high-fat diets, and they actually saw that PFOS exposure increased proliferation of the cell lines, and they did propose a mechanism through PPAR gamma that was linked to, I think, to immunosuppression or something like that. So it kind of reminded me what you were discussing right now. So I wanted to make sure that you guys had considered that study, because it links PFOS to PPAR gamma and to the immune system, particularly for prostate cancer, which is the second cancer. So breast cancer was the main one, as you know, that was the focus of study for the human studies, but the other cancer for which there are 3 studies that reported on PFOS exposure was prostate. And one did report an intriguing association that was positive with a positive trend. So I was curious about that study and whether you consider that one.
Could I ask you, what's the first author's name?
Emir, it's I-M-I-R.
So it
Yeah,
looks like maybe Dr. Sun has a comment or an answer to that.
Yeah.
Thank you, Dr. Loomis. I want to say 2 issues. One is a correction to what I said earlier about the historical control range for female rats, Dr. Eastman. Yeah, for female rats, the liver adenoma range should be 1.1 to 3.07%. So it's above 1 instead of the 0 to 0.29 that I said earlier. So I want to correct that. Thank you.
Thank you. I figured there was something wrong.
Yeah. The other thing is I want to answer Dr. John's question on the KC7, the human studies. I think the only one we've found is the one on natural killer cell activity in cultured human peripheral blood mononuclear cells. There's a decrease in the natural killer cell activity.
Thank you.
That's all for now. Thank you.
To answer Dr. Stern, I don't have in my notes about the INAR. I was just trying to log into the portal with the primary publications to see if that was in there. But you know, as far as the reason I called it strong was because of my experience with IARC in terms of the fact that we do have 3 different human cell lines that have increased proliferation and 3 human cell lines that have increased occurrence in S phase. And so, you know, the fact that, you know, we're trying to say, okay, well, in this period of time, we're supposed to take this body of evidence that's organized by the KCs, which makes it very easy to efficiently not overlook any of the evidence, but it's still more comforting to us to make a decision if we can, you know, tie these things together. From my perspective, when I think of estrogen receptor and being involved in proliferation, and then I see estrogen receptor dependent proliferation upon exposure to PFOS, and I also see the cell cycle phase being synced into that manner, and DNA methylation that's involved in cell cycle processes like the checkpoint RAD1 DNA methylation mark that I mentioned. To me, that, you know, tells a story, and it's not weaving in yet the oxidative stress, which can be, I think, sometimes a non-specific reaction. It need not be. I mean, clearly 8-oxo-DG is a very specific type of DNA adduct, and the fact that it came up consistently in 2 human studies with dose response, I think is quite strong also. But I'm going to look for this INER while other people talk.
Okay, let's see. Dr. Sandy has a hand up.
Yes, thank you. I'm pretty sure, and I'll ask Mung to confirm this, that we did not include that paper in our document, and it's probably not loaded on the FTP site, but if Mung can confirm that.
No.
I think that's correct, Dr. Sandy. We're talking about the EMIR paper, right?
Mm-hmm.
Correct.
Not.
Yeah.
All right, let's go on and see if there are any other questions or comments from the committee then. I'm not seeing any hands. Does that mean that you are all satisfied with what we've heard so far and ready to move on to public comments?
Dr. Lamerle has her hand raised.
Okay, thanks. I'm sorry I missed you. Go ahead, please.
Yeah, this is a question for you, Dr. Bush. I was wondering, I know that you had said when you were giving your overview of the animal tumor work that you recognize that there was some statistical significance, but it sounded like you were on the fence about whether or not it was biologically significant, and you wanted to hear more about the mechanism. So I was wondering if maybe you could revisit some, you know, that comment and really kind of reflect on some of the things that we've discussed. I'm just curious where you stand now.
Sure. Yeah, thanks for that, Dr. Lamerle. So the Covant study from 2002 was independent study, and then 10 years later, Butenhoff and that published the data. The first 3 authors including butanol for all 3M employees. So when looking at this data, kind of looking at it, taking it with a grain of salt, they don't mention the pancreatic cancer or tumors profile. So, thank you, OES staff, for generating that. In reading the public comments, though, I'm seeing this discrepancy, and Dr. Eastman kind of alluded to it, with respect to if PFOS has a major route through the liver, and if it is a peroxisome proliferator, maybe that is responsible for these liver effects that we're seeing. I think it is controversial, but if we don't include those, if we don't include that because of the controversy, the numbers don't look that exciting to me when looking at the Sprague-Dawley male and female mice. I was trying to quickly scribble down some of the historical tumor profiles that Dr. Sun was mentioning, and I believe you said hepatocellular adenomas were about 0 to 8% in Sprague-Dawley males.
Yes, 0 to 8% is the range.
Okay, so looking at table 6 in the in our document, the 0.5 ppm is at 7% of adenocarcinomas. The 20 ppm is at 16%. So, roughly double what we'd expect spontaneously anyway. Is that relevant? Maybe. I think I indicated in my comments that it's suggestive, but it's borderline for me in that regard. With the female liver cancers, we're not seeing this. For me, I'm not seeing a compelling dose response. Yes, I can probably statistically say that there's an increase with the 20 ppm treatment. But if this is a controversial area and we forget about liver effects altogether, then that really just leaves us with the pancreatic cancer, pancreatic tumors, excuse me, and the thyroid tumors. The thyroid tumors, as was indicated in the public comment from the American Chemical Society, right? US EPA has itself warned against the assessment of thyroid follicular cell tumors, and it may be considered less relevant to humans. So there's just not a lot of solid animal data that I can rely on. So coming back to Dr. Eastman's comments about the key characteristics, I do see bits and pieces there. And if that could be corroborated by the animal data, then a more compelling argument can be made. But I need more convincing, and some additional animal studies would certainly help. It would be good to have, teasing out whether there's a difference between the linear and the branched versions of PFOS and derivatives. And so at this point, hearing the mixed information about the the epidemiologic data, the bits and pieces with respect to the key characteristics, and I would say mixed or marginal animal data, for me, I am still not convinced when I weigh the evidence.
Thanks. Dr. Landolf, I'd like to hear from you now that you've had a chance to hear all this discussion, because at least initially you were a bit more positive about the animal data, I think. Would you like to revisit your comments? You're muted. Can't hear you. Still on mute.
Can you hear me now?
Yes.
Yeah. I'm looking at table 6 and I'm going to assume just for ease of the discussion that the denominator is the same. It varies a little bit, but for liver hepatocellular adenoma, they go 0, 3, 3, 1. So it goes 0, 3, 3, 1, and then 7. So it's crudely dose dependent. The 7 out of 43 at the 20 parts per million, that's the highest dose, is, let's see, they list that as statistically significant at p is less than 0.01. And the trend test p-value is 0.006. I don't feel compelled to throw that data away. To me, that data supports, the hypothesis that it is a carcinogen. The islet cell adenoma for pancreas, I indicated I would throw that away because it's basically the same number. That's a spontaneous result. The islet cell carcinoma for pancreas there's one tumor at 0, there's 2 at 0.5, there's 2 at 2 parts per million, then it jumps up, so it goes 1, 2, 2, 5, and 5. So it's kind of crudely dose dependent, it's not perfectly. And the trend test p-value is 0.048, so that looks like pretty good data to me. And then the thyroid data on page 48, table 7, it jumps up by a factor of 3. They only did a 0 dose and a 20 parts per million dose. And that went from roughly 3 out of 31 to 9 out of 29. So that's like a 3-fold increase, but it's positive. I could not throw that data away. And then the data on page 50, table 8, for the female rats, the adenomas in the liver go 0 at 0 dose, then they go 1, 1, 1, and 5. So that's kind of a slight increase, and then it's flat, and then it takes off at the 20. So there could be, some toxicity or something at the end there. I'm not sure, but that data is statistically significant at the 20 parts per million dose, and the trend test is p is less than 0.01. So that's pretty good data. The combined adenoma and carcinoma for the liver in the females go 0, 1, 1, 1, and 6, and that's statistically significant at 6, and the trend test is p is less than 0.01. The follicular cell adenoma for the thyroid goes 0, 0, 0, 2, and 1, so that's kind of a marginal increase there. The follicular cell carcinoma goes 0, 0, 0, 1, and 0. And if you add them together, it doesn't get you much more, and it's not statistically significant. And the fibroadenoma is statistically significant at the 0.5 dose, but not at the others, and the background is very high. So, and then the follicular cell adenoma in the female rats exposed to potassium PFOS goes at 0 doses, at 0 part per million, and jumps up barely to 1 at 20. So that's kind of marginal data. And then we had that data from the aflatoxin. And then the summary, if you look at that on page 126, the PFOS is positive in males and females for thyroid follicular cell adenoma and or carcinoma. You didn't get anything for PFOA. For liver hepatocellular adenoma and or carcinoma, it goes positive in the males and females for PFOS and PFOA. For pancreatic tumors, it's positive in the male and positive in the male and female. And for the testicular Leydig cell adenomas, it's positive in the male. And the mammary gland fibroadenoma, I think that data is marginal. They say it's positive in both females. So to me, there is data there that's positive, and I could not throw that out. I think the data more supports the hypothesis that it is a carcinogen than it does the data that it's not a carcinogen, because there's dose dependence and there's statistical significance. So I haven't changed my mind on that. I certainly would like, as Dr. Bush would and others would, to see more animal data, but we don't have it. We got to deal with what we've got. And but I can't throw that data away other than what I said I thought was marginal.
Okay, thanks. Dr. Eastman has another comment or question.
Thank you. There's a question more for you, Oiha, but I noticed for every one of these test doses, there's approximately 5 statistical tests being done on each dose. So, you know, there's always this concern about multiple comparisons and error, type 1 error that accumulates. Has OEHA looked at trying to control for multiple comparisons when they do these analyses?
I'll take a stab at that. So typically when evaluating cancer bioassay data, as is done usually by the National Toxicology Program and as well as by OEHA, we're looking at running pairwise comparison tests and trend tests for individual tumor types in certain organs. And we do not usually do any correction for multiple comparisons, because the assumption is you're testing something to see if it is causing an increase in tumors, and you're oftentimes the test chemical is going to target 1, 2, or 3 different target organs, and we're not running these tests on every single organ in the animal's body. Just
but they have been. The tests have been run on every single tissue. You only see the ones that were positive.
I'm not sure that's the case. We looked at the original study data, the Tomford et al. Report, and looked at tumor incidence without running any tests, and looked at which sites we thought there might be something going on, and then we applied the test. I wonder if Dr. Coliano has anything to add.
I think you pretty much covered it. I think that, yeah, there's a lot of tumor sites where there's no increase, and they're generally not reported in the published paper, and we're not running it on 40 different sites. We're running on what we see as positive. I think that's where the whole question of biological plausibility comes into play. And we look at the mechanistic data and key characteristics as indicators of biological plausibility of the tumor sites that we find to have an apparent increase.
But essentially, don't you mean then this is a post hoc analysis? Because you've already looked at the data and then you've chosen the ones to analyze based upon the results.
I think when there's only a couple of bioassays, that's pretty much the only thing you can do. It's not like this is something that's so well studied, and then we go out and design another study with a priori hypothesis that would affect the thyroid and the pancreas or some other organ.
No, I mean, the reason I brought this up is, it comes down to me, the liver tumors are what's driving this story. And they appear to be increased, both by a trend test at the high dose, by pairwise comparison, and they exceed historical control. So it makes some sense, but the p-values on some of these tumor instances are really pretty modest when you go into the pairwise comparisons. They're between 0.05 and 0.01. So they're not huge increases, but the consistency is there. I guess that's my thought.
No, I think those are good observations. This is something that's it does require some discussion, and we try to put together the different types of evidence and see where you come out. The statistics is one part of it. the plausibility is another part of it.
Makes sense.
Well,
I
thank
think
you.
if you're really concerned about those p-values, you can divide by the number of tests to get a sort of rough approximation of what you would have otherwise. Looks like Dr. Lamerle has a hand up again.
Yeah, I just addressing that there was numerous rodent studies that observed pancreatic tumors and well, they are all rats and male and females. It seems that the carcinogenicity of PFOS hasn't been studied in my weekend or in terms of multiple species, but you know, I didn't see any mechanistic data in the body of literature that we were provided related to the pancreas, but I did just type in PFOS and insulin into the internet, and there's about 4 or 5 different studies that have just come out in the last couple of years, consistently showing in rodent models that PFOS can modulate insulin secretion from pancreatic beta cells. I don't see anything addressing the pancreatic cancer aspect specifically, but it does appear that that the tissue, the pancreas, is a target of PFOS.
Thank you. Are there any other questions or comments from the committee before we close this part of the discussion?
Can I just ask, I recall on the OEHA slides this morning that there was indication that somebody had done some studies related to the genetic manipulation of the PPARs to try and address some of the doubts that are created about the relevance of PPARs in the context of PFOS-associated liver tumors, but I didn't see what the outcomes were or the details of that. Do you all happen to have that handy, OER?
Yeah, I can briefly describe the PPAR alpha related knockout mice study. I think in the PPAR alpha knockout mice, this one study reported PFOS-induced hepatomegaly, and one study reported upregulated genes in the liver of these mice. And also, yeah, and the EMIR study that we discussed earlier, PFOS activate human PPAR alpha in vitro and in this xenograft mouse model.
Thanks. Now, Dr. Crespi has a hand up.
Yeah, so I wanted to just a little bit relevant to the multiple testing issue. I think that you know, I think it's fine not to correct for multiple testing here. I think that we should think of the p-values as giving us some kind of rough evidence, and we should not draw a bright line at 0.05. That we need to think of everything in the totality and in the context. But so, like, for example, for this finding for in the male rats, the pancreas, the islet cell carcinoma at 0.048. You know, I wouldn't give that too much credence given the number of tests that have been done here. But thinking about the liver tumors where there are, the trend tests do have relatively low p-values, and also there's consistency across the two sexes. So to me, that, you know, that gives it that association more weight because of the consistency perhaps across the two sexes. And that's why I'm a little bit stuck on this one, trying to think about how relevant that might be, if that's translatable to humans. It's kind of where I feel a little bit stuck.
Would anyone like to comment on the question of human relevance?
Isn't it the finding at IARC that they consider tumors in rodents and tumors in people, and that even if they're not at the same site, they still consider it kind of carcinogenic one spot at a time in that sense?
It is, that's concordance between humans and animals isn't expected according to the IARC criteria.
Does that help, Kate? Dr. Crespi?
Yeah, I definitely. I mean, we don't, that part I understand. We don't expect if there's liver cancer that in one species, then, that means liver cancer. In the other species, necessarily it could just indicate carcinogenic potential. Yeah, I guess, yeah, I guess I just wonder more about, like, is there anything, special about this in this species of rats that would make us discount that or not?
Dr. Eastman, do you have a comment on this point?
Yeah, just quickly. Even the guidance within this committee is essentially, if it's positive in rodents, we go forward unless we have evidence to conclude it's not relevant in humans. So that's the issue in so much. And, I think these liver tumors may be caused through these mechanisms that are of questionable relevance to humans, but I don't think we have the evidence to conclude that. So that's the way I approach this.
Thank you. Okay, let's see if we can close this portion out one more time. Is there a question or a comment that we haven't heard yet? I don't see any raised hands.
No raised hands.
Okay, very good. Then that concludes the committee discussion and brings us to the public comment opportunity. So the way this will work is that we'll take comments from members of the public who've asked to speak. Those comments will be limited to 5 minutes each speaker. And to kick this off, I'll ask Dr. Martyr to show the public comment slide. So this is the housekeeping slide. So if anyone would like to make a comment, they can go to the URL shown on this slide and fill out a speaker request card. Some people may have done this already. Alternatively, you can click the raise hand icon on the Zoom screen if you would like to speak. So first we'll go to those requests to speak that have already come in by speaker request cards, and I'll ask Julian whether we have any speaker request cards, and if so, how many?
Yes, so we have 7 at the moment, and the first, if we're ready for that, is Andrea Ventura of Clean Water Action.
Okay, we'll go forward with that in just a moment, but I want to remind the other speakers that you have 5 minutes, and we'll take these speaker requests in the order in which they were received. So when one speaker finishes, Julian can announce the next one, and we'll just go ahead in that order. First speaker, you're welcome to go ahead.
Just a reminder to those speakers, they should have their name match that of the speaker card. So this speaker does not appear currently in our attendee list.
Okay, so the next is Jimena Diaz-Leyva of the Center for Environmental Health.
I am allowing Jimena Diaz to speak. You must unmute yourself first.
Hello, thank you for the opportunity to present comments. The Center for Environmental Health strongly supports the proposal to list perfluorooctane sulfonic acid and its salts in transformation and degradation precursors as known to the state to cause cancer under Proposition 65. We believe the weight of scientific evidence, including evidence of carcinogenicity in animals and mechanistic evidence of carcinogenicity, supports listing PFOS, its salts and precursors. And we believe the listing is very crucial to protecting public health because of the widespread occurrence, persistence, mobility, and potential to cause health harms of PFOS. Thank you.
Thank you.
Okay, the next card is Amber Lee Woodby. And not someone I'm seeing on the attendee list. So that takes us to Steve Risotto of the American Chemistry Council.
Who is present, and I am allowing you to talk.
Thank you. Can you hear me okay?
We can.
Yes. All right, awesome. Thank you for the opportunity to comment on the committee's consideration of carcinogenic evidence for PFOS. I'm Steve Risotto, representing the American Chemistry Council. ACC provided written comments in November. I would like to highlight a few points from those comments. As the committee has discussed, the data from epidemiology studies and cancer bioassays are mixed and sometimes contradictory. The finding in humans that appears to generate the most concern comes from the study of breast cancer among French women by Mancini et al. It is important to note that the association with estrogen receptor positive tumors was observed in only one of the 3 adjusted models applied by the researchers. The results from this model exhibit wide confidence intervals, however, that limit their interpretation. In the one available animal study, the strongest evidence is for liver tumors. As has been noted, evidence exists that these tumors result from a rodent-specific mechanism or mechanisms that may be of limited relevance to humans. This suggestion is strengthened by the fact that liver tumors have not been reported in the available epidemiology. In light of these equivocal data, OHIA's focus is directed at the evidence for the 10 key characteristics of carcinogens as a basis for the proposed listing. The 2017 publication by Becker et al. That applied the key characteristics to the data from high-throughput studies for over 200 chemicals evaluated by US EPA's Pesticide Office, however, found that the ability to predict cancer hazard was, quote, no better than chance, unquote. OHIA staff have themselves acknowledged that the key characteristics are of limited value for cancer hazard identification. While the key characteristics are useful for identifying and organizing data, the use of these characteristics does not meet the criteria for listing under Proposition 65. Given this information, the available data do not support a cancer hazard listing for PFOS. This is the conclusion reached by the European Food Safety Authority, Health Canada, and US EPA. Thank you.
Thank you. Is there a next speaker?
Yes, the next speaker is Suzanne Hume of cleanearthforkids.org.
Suzanne, if you unmute, you have been given permission to speak.
Hello, and thank you. Thank you so much to everyone here today. I just would like to say thank you to the fantastic OEHA staff. My name is Suzanne Hume. I'm the Educational Director and founder of cleanearthforkids.org. We're a nonprofit dedicated to children's health, public health, environmental and social justice, clean air and water. We ask you to please list PFOS as Prop 65. Studies have confirmed the human health risks of PFOS, especially for exposure through food and drinking water. The job of OEHA and the role of Prop 65 is to protect us. The American people are relying on you. And the American people do not need these studies in a neat package with a bow. Things are not always straightforward, but if they're causing cancer, if they're causing toxicity, in animals and humans, we need you to take action. We rely on you to protect us. When we talk about oxidative stress in cord blood in Chinese babies, it matters. When we talk about breast cancer, prostate cancer, pancreatic cancer, thyroid tumors, and liver cancer, it matters. We have deep concerns about PFOS causing oxidative stress and radicals that cue inflammation, strong associations, epigenetics, and the number of methylation studies, also demonstrated by firefighter study that Dr. Zhang shared, and deep concern about DNA methylation changes of cells in the cell cycle, relevant epigenetic data regarding the breast and alteration of microRNA and risk associations in ER positives. We have concerns about receptor mediation, mediation or mediated on ER alpha promoter activity, kidney and human breast cells, and increased proliferation of cells seen across human and animal studies, human cells being in S phase. We have concerns. Studies have shown increased mutagenicity, chromosomal effects, DNA damage, oxidation, DNA damage, increased ROS, RNS, and more. I'm a little nervous, I must say, presenting to this panel of experts and scientists. You've read the research and it's clear, and we've read this research as well. I'm so sorry that I'm the one presenting this data and not saying it as clearly as you would. We're very concerned about oxidative stress. We're very concerned about studies showing exposure to PFOS has problems with neurotoxicity, reproductive toxicity, immunotoxicity, thyroid disruption, cardiovascular toxicity, pulmonary toxicity, renal toxicity in laboratory animals, and many in vitro human systems. And so of course we're very concerned, and we're asking you to take action. We're very concerned about the human health risks and chronic toxicity, molecular mechanisms of PFOS. So as you know, PFAS really must be treated as a class. DTSC has already determined the regulation of individual PFAS is ineffective, and California treats them as a class shown by SB 343, AB 1200, and AB 1201, which were signed by Governor Newsom on October 5th. PFAS is a class and share many characteristics and toxicity. PFOA was listed under Prop 65 in September 2019 because it can cause birth defects and other reproductive harm. According to the EPA, both PFOS and PFOA are toxic to laboratory animals, producing reproductive, developmental, and systemic effects in laboratory tests, and with suggestive evidence of PFOS and PFOA causing cancer. PFAS are known as, PFAS are known as forever chemicals, as they're extremely strong and don't break down in the environment in our bodies, are linked to liver damage, thyroid disease, developmental reproductive problems, high cholesterol, obesity, immunity issues, hormone suppression, and several types of cancer. So today, and not very eloquently, I apologize, we are asking you to please take action to protect public health and add PFOS as carcinogenic, and consider adding all PFAS, PFAS as a class to Prop 65. Additionally, just a quick note, of course, we're very concerned about the history and present day of 3M, and also the American Chemistry Council and manufacturers as well of PFAS being so intimately involved with the regulation of these chemicals. So we're asking you to please take action. The American public relies on you. Thank you so much for your dedicated work. Thank you.
Thanks for your comment. Who's the next speaker, please?
John Bodaroff, also of cleanearthforkids.org.
You have been allowed to talk. You must unmute yourself. Thank you.
Thank you so much. My name is John Bodaroff with cleanearthforkids.org. I ask that you please list PFOs as carcinogenic under Prop 65. I also ask that all PFAs be treated as a class and added to Prop 65 as well. According to the EPA, both PFOs and PFOA, quote, are toxic to laboratory animals, producing reproductive, developmental, and systemic effects in laboratory tests, unquote, with, quote, suggestive evidence that PFOS and PFOA may cause cancer, unquote. PFOS and PFOA have a long history of harm. DuPont was sued in 1999 over PFOS and PFOA contamination. Court documents show DuPont and PFOA inventor 3M had secretly been doing medical studies on PFOA and PFOS for decades. 3M researchers stated in 1978 that PFOA and PFOS, quote, should be regarded as toxic, unquote. In 1981, 3M found that PFOA caused birth defects in rats. DuPont knew PFOA causes cancerous tumors in lab animals by the 1990s. In 2005, DuPont reached a settlement with the EPA for concealing their knowledge of PFOA toxicity. And a lot of these, this is all important because a lot of these manufacturers block studies. They make sure that these studies are not funded. So just because we don't have studies has nothing to do with the fact that these may cause cancer. There is evidence that PFOS are a health risk, just like PFOA and all the PFAS across the class. Please take action to protect public health and add PFOS as carcinogenic, and also consider adding all PFAs as a class to Prop 65. Thank you so much for your time.
Thank you. Who's the next speaker, please?
The last speaker card is from Evelyn of Clean Earth for Kids, but I'm not seeing that speaker in the list.
No. And as a reminder, you may rename yourself in Zoom if any of you have been called who have presented a speaker card but don't have a matching name. I am not seeing any changes in names in the list. Dr. Loomis.
Very good. Thank you. So now let's see if there are any raised hands.
There are no raised hands at this time.
And have we received any more speaker cards in the last minute or two?
We have not.
Okay, thank you very much. Thank you to all the speakers. That concludes the public comment opportunity.
We do have a raised hand from from Dr. Sandy.
Okay, please go ahead.
Thank you. I just wanted to take a moment to correct a misstatement by the commenter for the American Chemistry Council. OEA has not made any comments questioning the usefulness of the key characteristics of carcinogens. The commenter seems to be confusing two very different things: the key characteristics of carcinogens with a set of high throughput screening assays, the ToxCast and Tox21 assays. So as you know, the key characteristics of carcinogens were identified based on a comprehensive review of more than 100 agents classified by IARC as known to cause cancer in humans. And there's many sources of data, mechanistic data, that can inform those key characteristics, including data from humans, human cells, animals, animal cells, and cell-free systems, and other high throughput screening assays. And so in looking at the ToxCast and Tox21 high throughput screening assays, it's been noted by many people, including OEHA, that those assays, which were not designed to cover the key characteristics of carcinogens, don't fully cover them. And recommendations have been made that those ToxCast assays should be improved and to be a better set of information that might inform the key characteristics. That's all. Thank you.
Thanks for that clarification. Let's move on, if there are no more raised hands, to committee discussion and vote on the question before us. So first, before we proceed to a vote, I wanted to see whether committee members had any other comments that they would like to make. Dr. Landolf.
Yes, sir. Thank you. Yeah, I wanted to go over just briefly again I had discussed slightly, which is that the PFOS and the PFOA cause oxidative damage, the 8-hydroxydeoxyguanosine. There's data on immunosuppression. There's data that apoptosis is inhibited, senescence is inhibited, and the gap junctional intracellular communication is inhibited. So all these characteristics are consistent, and the fact that they cause transformation in Syrian hamster embryo cells and normal human breast epithelial cells. So I come out of a cell transformation background, and all these characteristics are ancillary data compared to the hard data like animal carcinogenesis, but they're all consistent with cell transformation and therefore carcinogenesis. So in my mind, I add this to the animal carcinogenesis data that's positive, and that moves me towards not a perfect answer, but an answer that's consistent with support for the hypothesis that these are carcinogens.
Thanks, Dr. Landolf. Any other comments from the committee? Go ahead, please, Dr. Stern.
I just want to add that there is one additional study that OIHA excluded from review for good reasons, because, you know, a priori they made a decision that they would include only studies that met certain criteria. One of them was measuring PFOS at the time of diagnosis or before diagnosis. This particular study is a case control study in Taiwan that measured PFOS at various times between time of diagnosis and after, yet they made sure to measure it before treatment. So the concern is reverse causation. That said, if there were indeed reverse causation, the more likely scenario is that it would bias the results towards the null, right? So that's kind of how we interpret that. In this particular study, they also found a positive association with breast cancer among ER-positive women. So even though we did not include that in our review, I keep coming back to that in light of all the discussions that we've been having, and I wanted to share that with the committee.
Thank you. Are there any other comments from the committee?
Dr. Lamero has her hand raised as well. Dr. Loomis.
Thanks. Go ahead, please.
Yeah, I just wanted to bring up the thyroid again. I was hoping that some of the public comments would help clarify some of my confusion about why we were supposed to discount the thyroid tumors in the rodents. But I do know that the thyroid in a systematic review of epidemiology studies, they found evidence of a positive association between PFOS exposure and TSH. And so, you know, I had summarized something I had found earlier on that level, but that does mean, you know, we've looked at numerous human studies that are seeing changes in thyroid hormone. I'm a little concerned about that, you know, that might be getting glossed over a little bit, since we're seeing lesions in the animals that are, I believe I remember reading that they were suggested to be rare, and it seemed like they were in both sexes. And then there's some evidence that this is operating in humans. So I just wanted to make sure that was known about the human study.
Thank you. Dr. McDonald has a hand up.
Yes, thank you. I really appreciate OIHA compiling all this primary source information. You've done a Herculean job as well. I did want to speak to the thyroid tumor, seeing we're on that topic. I wanted to make one point. With respect to male rats and thyroid tumors, there was an increase in adenoma, benign tumor at the 52-week recovery group, but there was no tumors seen in the 104-week group, which, as you would expect, in those to progress. I also wanted to point out that if you look at the follicular cell carcinomas, they actually decreased there's a dose-related decrease over all doses relative to controls. And so for me, I just don't see much evidence for male rat thyroid. Female rats, granted they are rare, but there were no statistical significance. So I'm kind of in the camp that, you know, these are suggestive but limited evidence. Thanks.
Is there anything else? So Dr. Crespi has a hand up.
Yeah, so I found what Dr. Stern said very interesting and relevant, and it makes me concerned that the HID may not have provided information on studies where exposure assessment was undertaken after diagnosis. And I just wonder whether it might have made sense to include such studies, considering that this is a chemical with a very long half-life that my understanding is it's not really metabolized in the body or well excreted. So like a reverse causation hypothesis seems very unlikely. So I wonder if excluding such studies might have been, might have led to us missing some relevant information.
Well, just to be clear, those studies that measured exposure at the time of diagnosis or afterward were included. They're reviewed in the section on breast cancer, but not in very much detail. However, the papers are available for the committee to review. So they are there.
Yes, sorry to interrupt. I'll just clarify that the study Dr. Stern mentioned, Tsai et al., and the title says it's a case control study, but it is of a cross-sectional design. So we have a list of studies that we initially identified, but we excluded, listed in the HID in the Appendix D, and we list the reason for exclusion.
Thanks for that. Okay, Dr. Crespi, does that address your question? Looks like Dr. Landolf has another comment.
Yes, thank you, Dr. Loomis. Dr. Stern, my friend and colleague from USC, you straddle both worlds as an epidemiologist and molecular carcinogenesis researcher. So my impression is that the epidemiology studies are somewhat insensitive. Is it possible that they're not sensitive enough yet to catch these materials, but the simpler things like the animal carcinogenesis, the gap junctional inhibition of intracellular communication, and assays like this could be sensitive enough to catch them, and the epidemiology just is not big enough and doesn't have the sensitivity to catch them yet? Is that possible?
Yeah, I can provide an answer, and then I would like for my colleagues, my epidemiology colleagues, Dr. Loomis and Reynolds, to also comment on this. But yes, it is challenging with the epidemiological studies because all of them did a one-time measurement, except for the Alabama cohort, which used a job matrix. The other ones did one-time serum measurement. So you are assuming that one measurement captures the typical exposure of those individuals that may have contributed to the development of cancer. So that's always a challenge in epidemiology. In spite of that, we do see some studies that show positive associations, others don't show anything, and others show positive associations that are not significant. So we tend to get concerned when we see that because we worry about confounding, we worry about particularly confounding by other PFAS, such as PFOA. So yes, it is tricky and it's not as clean as the experimental studies.
Thank you.
I don't know if that answers your question, but
yeah, it does. It does. Mm-hmm. Thank you.
I would just add that limitations of exposure assessment are probably the greatest limitation on the sensitivity of the studies that we have now. And that primarily relates to inter-individual variability in the excretion of these compounds. If it's true that half-life is relatively long, then perhaps within an individual, it doesn't matter that much when they're sampled, even though there's only one measurement in time, unless there are critical windows like we've discussed that are somehow missed by a single sample. But there may be quite a bit of variation between individuals and how they process the chemicals, and we really don't have any information at all about that. Anything else? Let's see, Dr. Reynolds.
I just wanted to piggyback on that comment that the one-time sample problem, I mean, some people have taken a look at that, and there seems to be fairly high correlations in studies which have had multiple samples in multiple periods of time. But the one-time sampling, one issue is the persistence of the chemical in the body. The other is the persistence of exposure, which in many of these cases is ongoing. So just to add that element.
Thanks, very good point. Let's see, Dr. McDonald, did you have your hand up again, or was that from before?
No, I did want to ask my epidemiology colleagues on the panel. One thing that I noticed in a number of the studies was the quartiles. The difference between exposure amongst one group to the next was really tiny, like 20% difference, 15–20% difference. And I just wondered if those are meaningful. I mean, they just seem like such a narrow range within the population that it's hard to break up into meaningful groups. Just wondered if that plays a role at all.
Anybody want to field that one?
So, Dr. McDonald, are you referring to the change from one quartile to the next?
Yes.
Yeah. Often when we see so ideally, if there's a causal relationship between an exposure and disease, we like to see that as quartiles increase, we see nice increases, and a test of trend will give us a significant finding. Now, when we don't see that, there could be multiple reasons. One of them is that there's residual confounding by something that we're not capturing, or that there's not a linear, we're assuming a linear relationship when we do those tests. The relationship does not have to be linear. We know that from other examples, right? In epidemiology, often exposures do not follow a linear relationship. There might be a plateau point up, and above that point, there's no further increase, or there might be all kinds of complicated relationships that we still have not figured out. So I personally take that into account in assessing causality, but we have to keep an open mind that there could be explanations for that. But the first thing that comes to my mind when I look at that is that there could be residual confounding. I don't know what others feel like, Dr. Reynolds or Loomis or Dr. Mack.
I do think your point about linearity is a good one, because we do see for all kinds of exposures, they're often not linear. And in epi studies, sometimes there's some variability in measurement, so that it's useful, rather than trying to take a continuous variable, to break it up into quartiles or quintiles to see whether or not there's some evidence of differences in extremes. So there are a variety of reasons, I think, for doing that, and that does help interpret the evidence.
So just to add on to that, one of the other challenges we have with sensitivity is that exposures in the general population tend to be quite low. And so that means whether you create quartiles or use a continuous exposure variable, well, the changes are indeed likely to be small. So what we would really like to have would be some occupational studies, of workers with higher exposures that would help to corroborate the evidence from the general population, but perhaps with greater sensitivity. And unfortunately, we just don't have those in this instance. Dr. Jung, comment, question?
Yes, just to follow Dr. Reynolds' comment on that one-time sample. I know Dr. Looms is the first one mentioned that. I thought I just add on that. For a lot of epidemiological studies, lots of times we may not even have any biological samples. So one thing I think at least the study has some biological samples and it did the exposure assessment. So that's, I would still put it as the positive point for that study. And the second, I think also for the chemicals, it's relatively stable. So it's not like some chemical, like, it could be, half-life is really short, then that would be a problem. So it's sort of on that sense. So I still think give a credit to the study. Another point I want to make is maybe just trying to make sure I express the previously when I was comment on the KC2, the genotoxicity of the PFOS. I think I want to do another, number one is a self-correction. I was saying even though it could be a weak genotoxic compound, but I think I just look over the data one more time, and I think even though some data is contradictory, but overall I think the in vivo data, both in humans and exposed animal, generally still pretty strong. So what I'm saying is, I was trying to say if PFOS can cause cancer, it may not just going through epigenetic pathway or genotoxic pathway, or could be epigenetic, or could also play the role in non-genotoxic pathway. So I don't think at this point which I could to identify if the PFOS is really a strong or weak genotoxic compound or not. So I want to take that back. Just try to get on the record.
Thank you. I don't see any other hands raised at this moment. So unless there are any burning last minute comments or questions from the committee, I would propose we move on to vote and decision. Is everyone ready?
Dr. Loomis, this is Carol Cummings. I just wanted to interject and remind the committee, if you're not comfortable making a decision today, it sounds like there's a lot of discussion back and forth and you probably understand it better than I do, but I just want to make sure that you know that you don't have to make a decision today. If you aren't comfortable with that, you can ask for, more data, you can table the question, or you can go ahead and vote. It's entirely up to you. Thanks.
So would it be appropriate then to ask if there is any proposal to table the decision?
Sure.
Is there a proposal to table the decision?
You have Dr. Bush with his hand raised.
Thank you. I'm not proposing that. I wanted to know, is abstention an option for us?
Yeah, this is Carol again. You can always abstain if you're not comfortable saying yes or no. It has essentially the effect of a no answer, however.
Thank you.
Mm-hmm.
All right, I don't hear any other proposal to table. So let us proceed to the vote. The question for decision is this: has perfluorooctane sulfonic acid, PFOS, and its salts and transformation and degradation precursors been clearly shown through scientifically valid testing according to a generally accepted principles to cause cancer. So on that question, you can vote yes, no, or abstain. And I'll go through and call for your votes in alphabetical order. Dr. Bush?
Abstain.
Dr. Crespi?
No.
Dr. Eastman?
Yes.
Dr. Lamero?
Yes.
Dr. Landolf?
Yes.
Dr. Loomis votes yes. Dr. Mack? We can't hear you.
Yes.
Dr. Mack votes yes. Dr. McDonald?
No.
Dr. Reynolds?
Yes.
Dr. Stern?
Yes.
Dr. Jung?
Yes.
Very good. So I count 1, 2, 3, 4, 5, 6, 7, 8 votes to list, 2 votes against listing, and 1 abstention. So that counts for a majority vote in favor of listing PFOS. So with that done, we'll turn to the next part of the agenda, which is a consent item updating the California Code of Regulations, Title 27, Section 27000, list of chemicals which have not been adequately tested as required. This is essentially a ministerial item, meaning that the committee is asked to affirm changes in response to submissions from the Department of Pesticide Regulation and the EPA. So I'll ask Julian Lichty now to present this item.
I'm sorry,
you, Doctor.
Julian.
Oh,
if you could
yeah.
just hold for a second. Dr. Loomis, when you asked for the vote and when you summarized it, you only said PFOS. And I'm just wondering whether or not you meant to include the whole group or that
Well,
just that one.
I meant to include the whole group because that was the question. So
Okay.
it's PFOS, its salts, transformation, and degradation precursors. I think we can't vote on anything else, right? Because that's the question in front of us.
Well, you could split them out. I mean, you've done that before when there was a group, but I just wanted to clarify that for the record that that's what you intended.
Well, I read the question as it was put in front of us.
Right.
So presumably that is what the committee understood, that they were voting on that entire group of chemicals named in the question.
Right.
I see heads nodding.
Okay.
Okay, so I think that's
Thank
what
you.
we've done. So if we're agreed on that and no one wants to revisit the vote, let's proceed with the staff presentation on the consent item.
All right, thank you, Dr. Loomis. So the slide, so the slide indicates the proposed change based on information received from the California Department of Pesticide Regulation, the removal of triethylene glycol detailed in the staff report provided to the committee. I will now turn this back to Dr. Loomis.
Dr. Loomis, you are muted. I believe you were reading the question, but you were muted.
Okay, sorry about that. So thanking Julian for that very quick but informative presentation. Again, this is a consent item, but it does require a formal vote on the following question. But before we go to that question, would any member of the committee like to comment or ask a question about it? Okay. Hearing and seeing nothing, the question that requires a vote then is, should section 27000 of the Title 27 California Code of Regulations be amended as indicated in the staff report? So again, I'll call your names in alphabetical order and ask you to vote yes, no, or abstain. Dr. Bush.
Yes.
Dr. Crespi.
Yes.
Dr. Eastman.
Yes.
Dr. Lamero.
Yes.
Dr. Landolf. Dr. Landolf, if you're voting, we can't hear you.
Oh, did you? Oh, okay. Yes. Sorry.
Thanks. Got you. Dr. Loomis votes yes. Dr. Mack. Could you repeat that? We couldn't hear you.
You're unmuted. It was a little garbled. Dr. Mack, just repeat.
Sorry, Dr. Mack, can you say it again? We just couldn't understand it. If you're having difficulty, would it be okay for you to type it into the chat?
Yes, sorry.
We heard you that time.
Thank you.
Thank you. Dr. McDonald.
Yes.
Dr. Reynolds.
Yes.
Dr. Stern.
Yes.
Dr. John.
Yes.
Okay, the vote is unanimous, so the change is affirmed. And now we'll move on to next item on the agenda, staff updates. We'll have updates on Proposition 65 listings, regulations, and litigation since the last meeting. So Julian Lichty again has the first presentation on listings and safe harbor levels. Please go ahead, Julian.
Thanks, Dr. Loomis. So since the committee's last meeting, we have administratively added a reproductive toxicity endpoint, developmental toxicity to the listing of bisphenol A, and we've added 2 chemicals to the Proposition 65 list as causing cancer. These chemicals are molybdenum trioxide and indium tin oxide. Next slide, please. I'll now move to the chemicals currently under consideration for administrative listing, which are perfluorooctanoic acid, PFOA, tetrahydrofuran, 2-ethylhexyl acrylate, methyl acrylate, and trimethyl propane triacrylate, technical grade. Next slide, please. Turning to safe harbor levels, since last meeting, 4 safe harbor levels have been adopted in regulation. No significant risk levels were adopted for p-chloroalpha-alpha-alpha trifluorotoluene, dibromoacetic acid, dichloroacetic acid, and trichloroacetic acid. Next slide, please. We have lastly proposed a safe harbor level for 1 chemical, 1,3-dichloropropene for the inhalation and oral routes. And I'll now turn things to Carol.
All right, Carol, please go ahead.
Right. Good afternoon again. For our other regulatory actions besides the safe harbor levels, we've been primarily working on safe harbor warnings for various chemicals, but we've also done a couple other things. As you may recall, last meeting, we were in the process of wrapping up some changes to the warnings for alcoholic beverages. It wasn't the content of the warning, it was the way to provide the warning that took into account that companies are now selling alcohol over the internet and through apps. So that became effective April 1st of 2021. We also have 2 regulations that are being considered for approval by the Office of Administrative Law. One is a regulation that would establish concentration levels for certain foods that are cooked or heat processed. And the first set of concentrations would be for acrylamide in those foods. And as I said, it's under review for hopefully for approval and filing. We also have at the Office of Administrative Law what we call tailored warnings for cannabis and THC products. There's 4 different versions of the warning depending on the type of, mostly the route of exposure, whether it's smoking, edibles, topical, that sort of thing. So we've adopted warnings and methods for providing warnings for those 2 chemicals. We are in the process of modifying our safe harbor warnings that we call short forms. We have longer warnings and we have the short form. And we determined that the short form warnings needed some modifications, including restricting the use of them to small products or packages, which was our intent, but the regulation didn't actually say that. And we are adding some requirements to name at least 1 chemical, even in the short form warning. So currently we're looking at the public comments on that and hope to have a decision about our next steps on that regulation soon. We have 2 other tailored warnings that we fairly recently proposed, one for acrylamide in foods, and that would of course cover those that aren't covered by the cooking or heat processing regulation. There will be foods that are above the concentration levels that we plan to adopt there, and so that's what this is for. And the same goes for glyphosate. We don't anticipate that most consumer products will need a warning, but there will be some, and so we have proposed a warning language for glyphosate. Okay, next slide. And just to update you briefly on litigation, our litigation list is getting shorter but more complicated. So we have the 2 cases in blue are actually in the federal courts. We haven't been in federal court very much under Prop 65, but we currently have these 2. I should say we means the state, because we are not as a WEHA part of these 2 cases, but certainly Prop 65, given that we implement it, we have an interest in both of them. As you'll see on the slide here, this litigation is about providing warnings for glyphosate exposures and acrylamide exposures from food. And these cases were part of the impetus for us to propose the specific warning language that I mentioned on the prior slide. The arguments in the cases are that providing a warning for these chemicals under Prop 65 would violate the company's First Amendment right against compelled commercial speech. Both of them, or at least parts of them, are in the Ninth Circuit Court of Appeal. Other parts are still in the trial courts. We have the very long-running Council for Education and Research on Toxics versus Starbucks case, which, as you may recall, has been on our list for some time. It's been about 10 years in litigation. Was recently decided by the trial court and is now on appeal in the California Courts of Appeal. It has to do with whether warnings are needed for coffee. As you may recall, we adopted a regulation last year, or maybe it was 2019, that determined that a warning is not required for coffee, even though there's acrylamide and other chemicals in coffee. But that's still, that case is on appeal. we have this case, the Physicians Committee for Responsible Medicine, I think was already on the list last time. And that is a request by this group for us to list processed meats as carcinogens under Prop 65, which we have declined to do. And we are in early stages of that case. We're negotiating discovery requests from PCRM. And lastly, we did resolve another long-running case of the American Chemistry Council versus OEHA, which had to do with the early listing of BPA as a developmental toxicant. After the trial court and Court of Appeal upheld that listing, I believe that it was the state Supreme Court declined to hear the case, and so now it's final, and we did relist bisphenol A for developmental effects. I think that's all I have, unless you have questions.
Thank you, Carol. Are there any questions?
Dr. Eastman has his hand raised. Dr. Lewis?
Yeah, I have a question. Carol, thanks for that overview. Over the years, we've been asked to hang on to paperwork that we have related to some of these cases. And I have forgotten which ones we're supposed to have. Could you send us out an email reminding us which materials we were supposed to be hanging on to? I mean, it's been years and years, so I never quite keep track of it. Thanks.
Okay. Yeah, I don't think you have very many, but we'll send you a list.
Thanks.
Any other questions? Okay, I can't see everyone on this screen.
No more questions, or at least no more hands raised, indicating questions.
Right. Okay, if there are none, then we will move to the very last item on the agenda. With that, I'll go back to Director Lauren Zeiss to summarize what we've done today.
Okay. Good afternoon. So the committee voted to add perfluorooctane sulfonic acid, PFOS, and its salts and transformation and degradation precursors to the Proposition 65 list. The vote was 8 yes, 2 no, and 1 abstain. So the chemicals will be added to the Proposition 65 list as known to cause cancer. Then the second item was the consent item, section 2700, and it was amended as indicated in the staff report, and the vote was unanimous. I guess I'd like to close by just thanking the audience, the public, for their participation in the meeting and for sharing with us their views. And also, we really do appreciate the written public comments we receive. It really helps with the whole body of evidence and helps the committee to consider it. So thank you very much for all of that input. Then I'd like to thank the committee for participating in the meeting today. Understand the amount of time it takes, taking time out of your very, very busy schedule. So we really very much appreciate it, and all the preparation that goes into this meeting. So thank you. And thank you, Dr. Loomis, for chairing the meeting today. We appreciate that. And then I'd really like to thank the staff for all the effort to put the document and this meeting together. And it was really gratifying to hear the comments on our hazard identification document. So thank you, staff, for all that effort and for all that went into this meeting. And with that, I'd like to wish you all a good and healthy and happy holiday, and looking forward to a very good 2022. And we will be seeing you in the next year. And with that, I'll turn it back over to you, Dana.
Thank you, Lauren. Well, I would just like to close by echoing all those comments. Thanks to the members of the public who took the time and effort to read the documents and comment and to listen into the meeting. Thanks to the committee members for all the work taken to review and work through the evidence. Really a very impressive job. And thanks especially to all of the OEHA staff for compiling these materials. It really was a heroic effort and a lot of information on this particular substance that was not at all easy to sort through. So thanks for all of that. In order to wish all of you a happy and healthy holiday, it's my pleasure to declare this meeting adjourned.